So, am I audible?
Yes, ma'am.
It is already 8:05, isn't it? Okay, good morning to all. I see only 17 have joined till now. That is around 16 have joined. It is already 8:05. I think we can start. Is there any connectivity issue?
No, ma'am.
No, ma'am.
In between, if there is any problem with the audio or video, representative can give me a call on the mobile. Now today's class, this is on basal ganglia. What I have projected here is a diagram which actually gives you an idea of where the basal ganglia is located. Now regarding basal ganglia, at the end of the 2 lectures of basal ganglia, what you should know mainly are the functions of basal ganglia and the disorders of basal ganglia, of which the most important one is Parkinson's disease. So now your specific learning objectives are to know the organization, which we shall be covering mainly today, organization of basal ganglia. Later on, we shall move on to the functions and the disorders of basal ganglia, mainly Parkinsonism. Now coming to the organization of basal ganglia. What is basal ganglia? Now it is related to the motor system, which already has been taken for you. Basal ganglia is an accessory motor system like cerebellum. So you have this descending tracts which are arising from the primary motor cortex with contributions from sensory cortex. These are descending down to the ventral horn of the spinal cord, and it is going to control your motor activity or movements. Along with this system, 2 areas which are required for coordinated motor activity. One is cerebellum, second is basal ganglia. That's why we call it an accessory motor system. Now, what actually does this basal ganglia contain? It's actually a group of mainly 5 deep subcortical nuclei. So these nuclei, which are collections of cell bodies, are present just beneath the outer layer of the cerebral cortex. That's why we call it as subcortical. It is below the cortical layer. It is deep within the white matter of the brain. Now, as you know, we have 2 cerebral hemispheres. If we take a section of the cerebral hemispheres, what is the location of white matter? What is the location of gray matter? Within the central part, you are having the white matter, and the peripheries contain the gray matter. Now, within the white matter of the forebrain itself, there are some collections of gray matter, and of these collections of gray matter, 5 deep subcortical nuclei together form the basal ganglia. Now, since these are present in the basal part of the cerebral hemispheres, and these are present on each side of the brain. Therefore, we call them as the basal ganglia, present or developed in the basal part of the cerebral hemispheres. Now, the first two components that we shall learn of basal ganglia, the most important ones, caudate nucleus and putamen. Now here, it is some new names that you're going to learn. There is no other way than memorizing these names. The caudate nucleus and putamen, these together are called as striatum. If you take a cut section of the cerebral hemispheres, the usual location of putamen is shown here. And almost beginning from one end of putamen as a C-shaped curved structure, you have the caudate nucleus. These together, caudate nucleus, putamen, are known together as striatum. The next part is globus pallidus. Now, the putamen and globus pallidus together, they form a lens-shaped structure. So these two together are also known as lenticular nucleus. Now, wherever such combination name comes, these may be asked for exams as short questions. What is striatum? It is caudate nucleus and putamen together known as striatum. What is lenticular nucleus? Putamen and globus pallidus together are known as lenticular nucleus. The fourth important component is subthalamic nucleus. It is below the thalamus, therefore it is known as subthalamic nucleus, also known as body of lice. The fifth important component is substantia nigra. Now these are the 5 major components of basal ganglia. Now here, the globus pallidus is again having 2 parts. One is globus pallidus externa, which is written as GPE, and globus pallidus interna, which is written as GPI. These are just the short forms which can be used. Globus pallidus externa, globus pallidus interna. The substantia nigra also has 2 parts. Substantia nigra pars compacta and substantia nigra pars reticulata. Now we are going to see a section of the brain which is taken from the coronal plane. Now when we say coronal plane, this exactly gives you an idea how the section is going to be taken. When you take such a section, this is the view that you're going to get, a coronal section, where this is the midline and you can see the 2 cerebral hemispheres. Now always it is important to have an idea of where the midline is going, because you can identify some important structures in relation to it. So for example, thalamus. Now thalamus is present on both sides and it's easy to understand its location near the midline. So here thalamus is shown, it's there on both sides. Just above the thalamus, you can see the lateral ventricles marked out. Now we shall try to understand the location of the 5 deep subcortical nuclei that we learned as basal ganglia. The first one is caudate nucleus, which is shown here on the right side. Next is putamen, which is shown in red. Just for understanding sake, we are shading it in red here. The next part is globus pallidus, which is having 2 parts. The first component that we have mentioned is globus pallidus externa, and more deeper to it comes the globus pallidus interna. Now, initially, we have mentioned that the caudate nucleus and putamen together, what are they known as? Caudate nucleus and putamen together are known as striatum. Whereas the putamen and the globus pallidus together are forming a lens-shaped nucleus. Therefore, this together are known as the lenticular nucleus. Next, subthalamic nucleus. Just below the thalamus, there is a small gray matter area called subthalamic nucleus. And just nearer to it, you have substantia nigra. Now, substantia nigra that also has two layers, substantia nigra pars compacta and substantia nigra pars reticularis. So now I think you have an idea where these 5 areas are located. Now here this is the lateral view where we are trying to understand the positions of the two components of striatum, which is caudate nucleus and putamen. Now when this is a lateral view, on the inner part, on the medial side, thalamus is shown. So it's easier for us to understand the location in relation to the medial structure, thalamus. So here you are having the putamen and globus pallidus together. And this is the caudate nucleus, which is having a head, a C-shaped curved part called body, which narrows down to form a tail, and the tail part ends in amygdaloid body. Now, if we take a horizontal view from the base part of the brain, here again, this is the midline, which is shown in red dotted lines. We are drawing the medial structure thalamus. In relation to thalamus, we shall just see the locations of the different parts of the basal ganglia. One is caudate nucleus, which is shown here. Next, laterally, there is putamen. More inner to it is globus pallidus. The first part is globus pallidus externa. Next comes globus pallidus interna. Now, between the caudate nucleus and the lenticular nucleus, you can see this V-shaped structure. This is the internal capsule, which is already discussed for you along with the pyramidal tracts when you're tracing. So you have the anterior limb of internal capsule, genu, and posterior limb of internal capsule. Now you have an idea where these structures are actually located. So we can very easily say that the nuclei of the components which are forming the basal ganglia, these are located lateral to thalamus and these are surrounding the thalamus. So whenever you draw, make sure you draw the midline, you mark the midline, mark out one prominent structure. It's easy to learn the relations in relation to that. Now moving on to few details of striatum. Corpus striatum was said to have two parts. This is one type of classification where striatum is mentioned as corpus striatum, and that is divided into two, neostriatum and paleostriatum. Neo means new. Neostriatum, also known as striatum, has two parts, which is caudate nucleus and putamen. I have just used the short forms because it is already discussed. Whenever you are using short forms in your answers, you have to explain it at least at one place, and then only you can use it. Now, if you consider phylogenetically, the two components of neostriatum or striatum, the caudate nucleus and putamen, these are more of recent origin phylogenetically. That's why we call them as new or new striatum. And functionally and structurally, they have similarities. So whenever we say striatum, it means caudate nucleus and putamen together. Paleostriatum, also known as pallidum, it refers to globus pallidus specifically. And this globus pallidus, it is the phylogenetically older part or the primitive part. The name itself, it is pallidum because when you take anatomical unstained sections, in this section itself, it appears paler. So pallidum, since it is pale, the name was given. So we have just discussed something regarding striatum. Now, in the striatum, or the neostriatum, the caudate nucleus that we mentioned, caudate nucleus, it's a structure. It is a C-shaped or comma-shaped structure which is having a head, a body which is curved as a C, and it ends as a slender part called the tail, which ends in amygdaloid nucleus. As previously mentioned, this is a lateral view in which we are seeing the caudate nucleus, the putamen and globus pallidus together, and inner to it, medial structure which is present is thalamus. The caudate, it's a larger part, it's having a head, body, and tail. Moving on to lenticular nucleus. Lenticular. It has the shape of a biconvex lens. It's a lens-shaped nucleus. Have a look at this diagram. This lens-shaped structure, it is called lenticular nucleus. The lateral component of lenticular nucleus is formed by putamen, and the medial component is formed by globus pallidus. Globus pallidus has two parts, globus pallidus externa and globus pallidus interna. Till this much, is the structure clear? Any doubts that you have?
No, ma'am.
Now, this is another view just to give you an idea of the location of the caudate nucleus, the lenticular nucleus, and how the fibers of the internal capsule are moving between them. So you have descending fibers which are going to move down towards the spinal cord, specifically to the ventral horn, which are going to control your motor activity. Which are the two descending tracts that we mainly have? Descending tracts. So from top to bottom. So cortex to spinal cord. That is a big clue, isn't it? It's corticospinal tracts. So when we say the pyramidal tracts, you have corticospinal and corticobulbar tracts mainly, which control movements. There is also role for the extra pyramidal fibers. Now, we had just seen the lenticular nucleus, and we have seen its parts, the lateral part being formed by the putamen, middle part is globus pallidus. These are not continuous structures. In this lenticular nucleus, in between, this lenticular nucleus is divided by a very thin layer of white matter. It's called the external lamina of white matter, which divides the lenticular nucleus into an outer darker part, the lateral putamen, and a medial part called globus pallidus. Even within the globus pallidus, there is an internal lamina of white matter which divides this globus pallidus into two parts. An outer segment called globus pallidus externa and an inner part called globus pallidus interna. So the lateral putamen, the outer part, it's darker in color. Pallidum or the globus pallidus, as the name itself implies, it is paler. This is the inner smaller part. Just a mention of the subthalamic nucleus of the body of lice. This subthalamic nucleus is also a biconvex mass of gray matter, but it is quite small. It is present just below the thalamus. And in relation to substantia nigra in the midbrain for the mesencephalon, it is present dorsal to the substantia nigra. We can understand that even in a diagram that we have seen. So just below the thalamus, you are having the subthalamic nucleus, and this is the location of the substantia nigra. It's easier to understand it in terms of the midline structure thalamus. You are having the subthalamic nucleus. Okay. Now regarding substantia nigra. Where is this substantia nigra present? I think you already will be having an idea that it is present in the midbrain. Now, what three components form the brainstem? When we say brainstem, which are the three components which are included along with it?
Midbrain, pons and medulla.
Midbrain, pons, medulla. So in the top part, midbrain, if you take a section, you can identify few parts. To understand that better, so this is midbrain, here you are having the pons and comes the medulla. If you just take a section of it and see, in the midbrain, we have a red nucleus, and here this portion is mentioned as substantia nigra. So it is this area that we are mentioning as substantia nigra. Now substantia nigra, this appears darker in sections as the neurons contain pigment neuromelanin. There are two parts to substantia nigra. One is the substantia nigra pars compacta, and this is the more dorsal part. Now the pars compacta of the two sides are actually continuous across the ventral tegmentum. So there is some cross connections or continuity between this substantia nigra pars compacta of both sides. If we look at the type of neurons which are present in the substantia nigra pars compacta, majority or 75% are dopaminergic neurons or neurons which secrete the neurotransmitter dopamine, and 25% are cholinergic neurons, neurons which are secreting the neurotransmitter acetylcholine. So mainly it is dopamine which is produced in the substantia nigra pars compacta. The substantia nigra pars reticularis, this is the more ventral part of substantia nigra, and it is interesting to note that superiorly it is continuous, having connections with the globus pallidus. And most of the neurons which are present in the substantia nigra pars reticularis, these are GABAergic neurons, or the neurotransmitter produced here is GABA, which is an inhibitory neurotransmitter. So now we have an idea of the different components of the basal ganglia. Next, we shall move on to the connections of basal ganglia. Now, generally connections are mentioned in terms of afferent or the sensory inputs and the efferent or the motor output. In terms of basal ganglia, we shall also discuss some interconnections as projection fibers or projections of basal ganglia. First, we shall see the afferent connections or the sensory inputs to the basal ganglia. All the sensory inputs or the afferents mainly reach the striatum. So what does striatum mean? The two components of basal ganglia, which are caudate nucleus and putamen. So the first afferent that we need to know, this is the corticostriate projection. So from the cortex to the striatum, inputs are reached. From which all areas of cortex? From the premotor area, supplementary motor area, somatosensory cortex, especially from the layer 5. The second type of afferents to the striatum, it is thalamostriate fibers. So the afferent or sensory inputs are coming from the thalamus to the striatum. Specifically in thalamus, it is from the intralaminar nuclei and the centromedian nucleus of thalamus that the afferents are coming. And they reach the striatum. The third type of afferent, it is the nigrostriatal projection. Nigro. So from the substantia nigra, but here we have to be careful that it is the substantia nigra pars compacta which is sending the afferents to striatum. So here the three afferents to striatum, one from cortex, one from thalamus, one from the substantia nigra pars compacta we have mentioned. Now this is just a schematic diagram. Suppose you want to draw it schematically, you draw a midline, draw the midline structure thalamus. This is not an actual diagram, this is a schematic diagram, just for understanding part. And the cerebral hemispheres will be there, and we are mentioning the cerebral cortex here, caudate nucleus. And this is an enlarged view of the lenticular nucleus. It is out of proportion. So the lateral part, you are having the putamen, and you are having the external lamina, the internal lamina of white matter, which divides the lenticular nucleus into the lateral part, putamen and globus pallidus. You have globus pallidus externa or the external segment, globus pallidus interna or the internal segment. Actually, it is easier to remember it this way, that the globus pallidus internal segment is interior, so it should be placed more medially. Just below the thalamus, you have the subthalamic nucleus. Then to substantia nigra, you have two parts, substantia nigra pars compacta, substantia nigra pars reticularis. So the afferent connections that we mentioned, from where all inputs are reaching the striatum, from the cerebral cortex, from the thalamus, from the substantia nigra pars compacta. The idea of putting this schematic diagram is to just to give you an idea from where all inputs are converging onto the striatum. So here, though it is to the caudate nucleus and putamen together, mainly what is shown for ease of representation is putamen. Now from cortex to the striatum, when the afferents are reaching, it is mainly excitatory neurotransmitters which are released at the endings, which may be glutamate or acetylcholine. From the substantia nigra pars compacta, the afferents which are reaching to the striatum are mainly dopaminergic, and these are excitatory. Now the effect of dopamine, whether it is stimulatory or inhibitory, depends on the type of receptor, which we shall discuss later on. Now, there are also afferents reaching the striatum from certain brainstem areas. At least few names to it that you should be knowing. Raphe striate projection. From the dorsal raphe nucleus, these areas of the midbrain and pons, afferents are reaching the striatum. And here, the neurotransmitter is mainly serotonin. From the pons pedunculo-pontine nuclei, afferents reaching to the striatum, these are called pedunculostriate fibers. PPN, this is a short form for pedunculo-pontine nuclei. Also, from the pontine locus ceruleus, afferents are reaching the striatum. These are called locus ceruleus striate fibers. Mainly it is norepinephrine, or these are noradrenergic fibers. So in effect, if we consider the afferents from the brainstem to the striatum, the neurotransmitters are generally serotonin, norepinephrine, or enkephalin. Whereas if you consider the afferents from the cortex, it is mainly glutamate, acetylcholine. If it is from the substantia nigra, it is mainly dopamine. Even if it is projections from the thalamus reaching the striatum, the afferents from thalamus to striatum, it's mainly excitatory, where the neurotransmitter may be glutamate or acetylcholine. So we have just finished the afferent or sensory inputs which reach on to the striatum. Now the projections which are coming from striatum. From the striatum, the nerve fibers are going to convey the information further on within the basal ganglia nuclei. So from the striatum, there are projection fibers going to the globus pallidus externa and globus pallidus interna. These are called striatopallidal fibers. Striatum to globus pallidus. And here the neurotransmitter is mainly inhibitory GABA. Now globus pallidus interna is given more importance because mainly efferents which arise from the basal ganglia, they arise mainly from the globus pallidus internal segment or globus pallidus interna. And from here, the efferents, they go to the cortex, they also go to the brainstem nuclei and influence the motor activity. The other type of projections from striatum, from the striatum, the fibers which go to the substantia nigra pars reticularis. These striatonigral fibers, at their endings, they release the inhibitory neurotransmitter GABA. So now we have seen two projections from the striatum to the globus pallidus to the substantia nigra pars reticulata or reticularis, and both of these are releasing inhibitory neurotransmitter at their endings. Now, if you consider the efferent or the outputs which are arising, outputs of the basal ganglia are mainly arising from globus pallidus. The efferents or the output mainly reach to the thalamus. Now, mainly from the globus pallidus interna, specifically to the thalamic ventroanterior, ventrolateral, and also centromedian nucleus of thalamus, the outputs are reaching. And GPI or the globus pallidus interna, it releases the inhibitor neurotransmitter GABA. So GPI is going to have some inhibition or inhibitory effect on the thalamus. These efferent fibers going from the globus pallidus interna to the thalamus, these are called thalamic fasciculus or ansa fasciculus. Now from the thalamus, thereafter nerve fibers arise and these are projecting to the cortex. So from thalamus to the premotor, prefrontal and the supplementary motor cortex, to these areas neurons will go, and at the level of cortex, the neurotransmitter released at the nerve endings, it is glutamate. So there is going to be excitation of the cortex. So when we consider the efferents mainly from the globus pallidus interna to the thalamus, there is inhibition because of GABA. Neurons from thalamus finally reach back to the cortex and there is excitation occurring there because of glutamate. Now we are taking the help of this schematic diagram again. We mentioned some projection fibers from striatum to globus pallidus internal segment or the globus pallidus external segment. Here the neurotransmitter is GABA, which is inhibitory. From striatum to the substantia nigra pars reticularis also we mentioned the fibers are inhibitory because of GABA. Now the afferents that we mentioned till now, efferent fibers we said mainly are arising from the globus pallidus interna. The output, so since the output from basal ganglia is going to influence motor activity, it is of most importance to understand how the efferents are having an effect on motor activity. GPI or globus pallidus interna the nerve fibers are reaching to thalamus. And here, the neurotransmitter release is GABA. So globus pallidus interna inhibits the thalamus. From the thalamus, the nerve fibers project back to the cerebral cortex. And at this level, the neurotransmitter is glutamate, which is excitatory. I hope the first efferent or the output is clear here. We shall move on to the other efferents or the outputs that are arising. One is from the subthalamic nucleus. From the subthalamic nucleus, nerve fibers, they project to substantia nigra. And from the substantia nigra, there are three routes which are going to be taken from the nerve fibers. What are these three routes? From the globus pallidus, either the nerve fibers directly reach the substantia nigra, or they relate to the subthalamic nucleus and then reach to the substantia nigra, or they reach via the brainstem nuclei. These are the three different routes taken. There are also some different outputs from the red nucleus of midbrain, where the fibers from the globus pallidus, they relate to the red nucleus, and from the red nucleus, rubrospinal tracts arise. This shows there are a lot of cross connections present within the basal ganglia, and then projections or efferent outputs are reaching to the brainstem and to the cortex. Now this makes things a little confusing. We shall try to make it more simpler. We have already mentioned the output or the efferent fibers from globus pallidus interna reach thalamus, the ventroanterior, ventrolateral nuclei, and also the centromedian nucleus of thalamus, where the neurotransmitter is GABA. GABA is inhibitory. The output from the thalamus, the nerve fibers which are arising, they reach back to the cerebral cortex. And here, the neurotransmitter being glutamate, it has an excitation of cerebral cortex. Now, this is desirable because cortical neurons then have to stimulate the motor activity by influencing the motor neurons that you have on the ventral horns of the spinal cord. Now, the basal ganglia efferents, there are also efferents arising from the globus pallidus to the red nucleus. From there, the rubrospinal tracts arise. If you consider the fibers from the striatum to the substantia nigra, from striatum to the substantia nigra, pars reticulata efferent fibers arise. Not only that, from the substantia nigra pars reticularis, there are also small fibers arising to the nearby thalamus, efferents reach there. Substantia nigra to the brainstem reticular fibers, there are efferent fibers. From the reticular formation, the next nerve fibers which relay the information down are the reticulospinal tract. From substantia nigra, nerve fibers also go to the midbrain superior colliculus, and from there, tectospinal tracts carry the information. Also from the substantia nigra to the habenula. Now, if you just look at these efferents, a large number of efferent fibers are arising. Now, the efferents from basal ganglia, these are going not only to the brainstem areas like the red nucleus, to the brainstem reticular formation, the superior colliculus. There they influence these nuclei, and the next neuron which forms a descending tract then influences the motor activity of the corresponding part. It may be by the rubrospinal tract, or the reticulospinal tract, or tectospinal tract. All of these, as you know, are components of the extrapyramidal systems. So this cross connections give you an idea that the pyramidal and extrapyramidal tracts, though they are primarily controlling movement, the accessory motor system basal ganglia is sending efferents to these areas and it has an influence on the control of movements. We shall just look at how the cross connections are there within the basal ganglia. We shall run through this flowchart once again. We already mentioned that from the cerebral cortex, there are afferents reaching the striatum. These are mainly excitatory. Not only glutamate, but acetylcholine is neurotransmitter involved here. Even from the thalamus, there is excitation of striatum. Again, glutamate, acetylcholine. From the substantia nigra, pars compacta layer, afferents reach the striatum. Dopamine is released and specifically acts via D1 receptors, and these are having a stimulatory or excitatory effect on the striatum. From the striatum, nerve fibers then project to globus pallidus externa. Here, the neurotransmitter released is GABA, so there is inhibition of the globus pallidus externa neurons. If there is inhibition here, there will be less transmission across the nerve fibers or the tracts which are arising from here. That's why a thinner red line is shown here. There is inhibition of globus pallidus externa. So less output arises from the globus pallidus externa to the subthalamic nucleus. Here again, the neurotransmitter released is inhibitory GABA. This is the subthalamic nucleus. From the striatum, directly also fibers reach to the inner part, the globus pallidus interna. These are also inhibitory because of the neurotransmitter GABA. In short, we can mention that striatum to globus pallidus externa or interna, there is an inhibition via GABA, gamma aminobutyric acid. Now, from the subthalamic nucleus, small neurons or the nerve fibers are coming, projecting to the globus pallidus interna. These are stimulatory or excitatory since the neurotransmitter released is glutamate. So, subthalamic nucleus stimulates the GPI. Now, when there is stimulation of the globus pallidus interna, the output from the globus pallidus interna will increase. So there is inhibition of thalamus. The neurotransmitter being released is GABA. So from the thalamus, the output neurons arising, reaching back to the cortex. These are excitatory to the cortex, but normally these are kept reduced. Whenever you are planning a motor activity, then only, or when you want to perform a motor activity, then only you require the cortical neurons to be stimulated. From the striatum, you also have efferents going to the substantia nigra pars reticularis, and those are inhibitory because of the neurotransmitter GABA. Now, this entire complicated diagram tells you the net interconnections of the basal ganglia neurons. What can be said regarding the internuclear connections? When the striatum it is projecting to the globus pallidus, there is mainly inhibition of globus pallidus externa and interna because of the neurotransmitter GABA. The striatonigral fibers which project from striatum to the substantia nigra pars reticularis, they are also inhibitory because of GABA. Afferents reaching the substantia nigra from the pars compacta, substantia nigra pars compacta to the striatum, the afferents which are coming via D1 receptors. When dopamine is active, it is having a stimulatory or excitatory effect. Globus pallidus externa, its direct influence on the subthalamic nucleus is inhibitory via the GABA. If you consider the effect of subthalamic nucleus back to the globus pallidus interna, it is stimulatory. It also gives stimulatory fibers to the globus pallidus externa and stimulatory fibers to substantia nigra. And that stimulation is via glutamate. Substantia nigra pars reticularis also gives small projections to thalamus, but it is inhibitory because of GABA. So the entire cross connections, if you put it in a single diagram, it becomes too complicated. One of the most important things that you have to note regarding this flow chart is the output which is arising from the globus pallidus interna. This is going to have a main influence on the thalamus, and from thalamus, the neurons are going to influence the cerebral cortex. Now, what is very briefly shown here is also some fibers reaching from the subthalamic nucleus to the pedunculum pontine nuclei, and from here to the brainstem neurons. Now, from these brainstem neurons, the efferent fibers finally go to control the motor activity. So, the extrapyramidal fibers are also having a role in influencing the motor activity. How will cortex influence the motor activity? The descending fibers from the cortex to the spinal cord, anterior motor neurons. That is the corticospinal tracts. To summarize the connections of the basal ganglia, what is most important to know? The main inputs which reach the basal ganglia are reaching the striatum, and the inputs are coming from the cortex, the corticostriate fibers, inputs coming from thalamus, thalamostriate fibers, and also inputs coming from the brainstem. What about the main output? The main output from the basal ganglia, it is via GPI or globus pallidus interna, and also from the substantia nigra to the thalamus. Keep in mind, the information from thalamus via neurons is finally sent to the cortex. Now, from where did this actual inputs start? From the cortex. Literally, we can say the information via neurons is reaching back to the cortex, and there is a sort of loop or a feedback which is occurring. Inputs come from the cortex, they influence the motor activity, the inputs are reaching back to the cortex. So not only the pyramidal, but also the extrapyramidal fibers are also influencing the final control of motor activity. So in effect, we can say there are motor loops. When we say motor loops, what forms the loop? Neurons bringing information from the cerebral cortex, they reach the striatum. From striatum, the inputs reach globus pallidus interna. From the globus pallidus interna, output neurons project to the thalamus. From thalamus, neurons reach back to the cortex. This reaching back to cortex completes the loop. So there is a primary feedback loop occurring. We can mention two major neuron circuits which are called putamen and caudate circuit. Now these circuit diagrams you can look up in the textbook of Guyton. Good diagram is given for that. When we say caudate circuit, what you need to know, what is the main function of this caudate circuit? It is the control of motor activity, but a cognitive control. Cognition refers to your thinking process. So whenever you are going to think and plan out an activity, that component is done by the caudate circuit. Also, the timing of a movement, how rapidly you are going to perform a movement, or scaling of a movement. Say, for example, how large a movement should be. When you write a letter as small or when you write it as big, how large a movement you have to move. All these control is occurring via the coordinate circuit. So this is a diagram just taken from textbook of Guyton, which shows the coordinate circuit. And this seems like a complicated diagram. It's easy to understand it this way. From the cerebral cortex, inputs reach the caudate nucleus. From the caudate nucleus to the globus pallidus internal segment. From there to the thalamus. From thalamus, information loops back or returns to the cortex. This feedback forms the caudate circuit. And what is the main function of this caudate circuit? It has a role in the cognitive control of motor activity, which is the thinking process of brain. Also, it has a role in control of eye movements. The caudate circuit, cognitive control of motor activity, also control of eye movements. Whereas putamen circuit, it is for the control of the skilled movements that you do. When you learn it, you do it consciously. Thereafter, once you have learned an activity, you do it subconsciously. Say, for example, writing the letters of an alphabet, you don't have to think where you have to move. It comes automatically once you have learned the alphabets. Cutting a paper with scissors, or a hammering a nail, passing a football, throwing a baseball, all these skilled activities occur subconsciously. Even vocalization as you speak, the different control movements of the eyes, all these are occurring subconsciously, and this is via putamen circuit. This diagram is also from Guyton. Putamen circuit, where inputs from cerebral cortex reach to the putamen. They are not going to the caudate nucleus, they are bypassing caudate. From putamen, they go to the globus pallidus interna, to the thalamus, back to the cortex. This is the simplest explanation for putamen circuit. Its function is for the motor control of the body movements. Now, the skilled movements, they are going to be controlled subconsciously. You are not aware of it. Now, please read up one of the standard textbooks for basal ganglia. We shall continue tomorrow with the rest of the basal ganglia. Until you have read up the circuits, you will not have a good idea of it. Any doubts you have, you can please ask. Please read up. There should be a lot of doubts coming. Then only we can clear it. Today, any doubts that you have? What we have covered till now is the normal circuitry. Once we understand the normal, then only we can move on to the disorders, which we shall be taking tomorrow. So please keep reading. Any doubts that you have, you can ask.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you.